unfortunately, the brain is not it's one big problem.
No, I realize that.
It's a pleasure to introduce, and I hope soon meet, David Sussillo. I wish I'd met you. Even though I don't know you, I'm going to give you a 2-second introduction. Okay. There is, I think David shows a problem and a solution. The problem is a very selfish problem for many of us in the room, actually, that extremely well-trained neuroscientists go to machine learning, and it's impossible to, losing to neuroscience wonderful minds and impossible.
Deep minds.
Impossible to hire people in this field to do the day-to-day work of data analysis and modeling and theory. But the wonderful solution of someone going off and doing, groundbreaking work in machine learning and applying it to neuroscience. So hope to meet you. Thank you.
All right.
And I rumor that you might be coming back.
This talk is being recorded. All right, so it's an honor to be here. So today I'm going to talk about inferring single trial neuropopulation dynamics using sequential autoencoders. And one of the pleasures of going near to last is that so many of the preliminary concepts have been already discussed and debated and introduced. So that's a nice thing. Let me at the outset acknowledge some collaborators, in particular Chetan Pandernath, who is now an assistant professor at Emory. He's really done the really hard and careful work of applying this technique to a number of data sets. And honestly, we couldn't have done it without him. Also, Dan O'Shea, who's I guess a postdoc in Krishna's lab, and Jasmine Collins, who is a PhD student here. And she was instrumental in helping us vet this technique on a bunch of simulated examples where we understand what the ground truth and we kind of know what's going on. All right, so I learned yesterday that we have some differing opinions about Mars, so I changed my outline. So we're going to introduce the 3 levels of Mars analysis, and for the first hour we're going to talk about computation. And then the second hour we'll dive into the algorithms, okay? And then finally, the last hour will be implementation. But you may or may not know, actually, there is a level 4 for the aficionados. All right. Okay, no, for real. What we're actually going to talk about is what has been bandied about is a dynamical systems hypothesis. And this is used in a certain group of people who think about motor cortex and prefrontal cortex. And I'll define what we mean by what I mean by this and how it motivates a data analysis technique. Okay. So it leads to this technique which we call LFADS. And I'll give a bunch of results that are basically improved decoding on single trials. We can study the single trial dynamics. We can use this technique to sort of stitch together data sets that are recorded on different days. And maybe the different days' data can synergistically improve the fits. And so we might talk, if we have enough time, about inferring inputs to neural circuits using this technique. And also oscillations, which seem to have come up a couple of times. Okay, so this is really a single talk with a few sub-pieces, and depending on the level of engagement during the talk, we may or may not end up finishing. Okay, so the hypothesis is that, let me see if I can, great. So the hypothesis is neural computation is explained by dynamics. So the evidence for dynamics in the brain is overwhelming. So the question is, what do we mean when we say that neural computation is explained by dynamics? And there is a recent literature about trying to define what we might call a low-dimensional subspace in which some abstract dynamics are occurring, and that these abstract dynamics are what these neural circuits are sort of actually doing. But so what do we mean by this? Let's see if we can explain it a little bit more. So we have our neural circuit here that we're going to study, and we look at the spikes of each neuron for K neurons. And so we just think of the population state. What are we actually studying here is this population state, which is just the bin spike counts of this neural circuit in some kind of operation during some kind of behavior. Okay. And then we have a sort of dynamic by just binning the spikes and moving those bin spikes through time. And so just to put a couple of different examples, more recently in the literature, we have, for example, low-dimensional dynamics in C. elegans, low-dimensional dynamics for contextual integration in the prefrontal cortex, and also rotatory dynamics in motor cortex when it comes to explaining reaches. So this data set is going to figure large into the talk I'm giving today. So let's jump in here. And well, this is, I think, the third time this slide has been thrown up by different speakers. So we have a neural state space, and we're going to take these bin spikes, and we imagine that there is a dynamics that is on some low-dimensional, in this case it looks like a subspace, but low-dimensional manifold. And we think of this as the state of the circuit, and that unlike, for example, the way that people think about the feed-forward networks and object recognition, we're not thinking about representations here so much as dynamics is what we're highlighting. And so, yeah, for example, for an animal that is making sort of complicated reaches through this maze-like structure, and here are some example reaches here in the XY plane, you get this dynamics. And so that's what we're about. We're trying to understand this. And so what do I mean when I say dynamical systems? I mean that this lower dimensional state has an initial condition, and that there is a dynamics that is dependent on the circuitry of that area, and that you have a vector field and you just integrate, and that different conditions do different things. Right? So in this world of an autonomous system, which is pretty simplified, we have the dynamics that we'd like to learn, and we have an initial condition, sort of a per condition, initial condition that we'd like to learn. And that would completely describe these dynamics.
But there's a signal, there's an input to the system, right?
That's right. So that is something that we will address. But for the majority of the talk, I'll be looking at the autonomous case while fully acknowledging that this is a simplified view. Totally. Okay? And so that's what I mean.
Autonomous means no input?
Autonomous means no input. Yeah, the full, so just to give you the full story, the LFADS architecture also has inputs that can be inferred so that we can do the non-autonomous case. But what we discovered in review is that if introducing the inputs early causes extreme doubts about what the dynamics are doing. And so I introduce it now later so that there can be no confusion about the dynamics explaining what the results that you see.
But I don't understand. You observe a very small part of the neurons, K neurons out of who knows how many, which are all connected to each other.
That's right.
How can you, what justifies the assumption of that these K neurons have autonomous dynamics?
So I'm not sure it's entirely justified a priori. But when one models these data with an autonomous system, one finds a very good match to the data. And so,
ignoring the rest of the network.
basically, yes. On top of, yes, essentially. And on top of this, the sort of very widely observed result that the dynamics of potentially due to the simplicity of the behavioral or the experimental setup, the dimensionality of the dynamics tends to be much lower than the number of neurons in the area.
So essentially, there's just like saying that what is the minimal k which actually justifies this?
That would be a theoretical question that I won't answer.
So I briefly mentioned this yesterday, which is that if you increase the number of you record from 200, but you say what happens if I keep adding? It keeps getting better and better. You get to 100, and then it sort of saturates. And by 200, you're not actually getting any more information out of the state, of the brain state. So it's telling you that, in fact, there's a lot of redundancy, right?
Right.
It were factors.
Too much redundancy.
Yeah.
A surprising amount. That's right. Shocking, in fact.
Given the experimental
Given the experimental setup, that's right. And as was mentioned previously, more recent recordings out of the Carandini lab are demonstrating very, very high dimensional data in V1. Okay, so this whole world, this whole story about dynamical systems explaining, for example, motor cortical data makes sense for conditioned average data. And so that's really what's been done. The condition averaging has been used to sort of get rid of some of the spiking noise and perhaps some of the other variability that's in these circuits. So the question is, well, what happens when you, and this is indeed the challenge that we're addressing today, what happens when you try to do this type of analysis on single trials? Well, if here I've showed you this rotatory plane, which was found using a technique called JPCA. If you run the JPCA technique on single trial data, now in this case we've taken a Utah array, so I think in this, maybe even two, so about 200 channels or so, it's a disaster. It just doesn't work. It doesn't make any sense on single trials. You can't see through the noise of it. And so that's what we're going to do. And so now I think it's useful to sort of outline what we've been talking about here with respect to this dynamical systems hypothesis in and of its own right. But this hypothesis also motivates a data analysis technique, and that's what LFADS is. LFADS is an embodiment of this hypothesis. And so let's now operationalize this hypothesis into a data analysis technique. So we have here our spikes through time, and so I'm really thinking now of high-dimensional spike trains that are being recorded. I'm really thinking towards the future of what neuroscience will be, and for many labs, the future is now. So we think of these spikes as having a sort of, each neuron has a firing rate, right? And you can get the firing rate by condition averaging, but you could imagine there's this thing called, it's an abstraction, but this thing called an instantaneous firing rate for each neuron. And again, we further imagine that these firing rates are composed of a lower dimensional set of temporal factors, right? Okay, so what we're arguing is that this all comes from a dynamical system that's nonlinear, and that what you'd really like to find is the code or the initial condition to generate the data that you've observed, right? And so LFADS is a sequential autoencoder. This is the decoder or generative model of LFADS. Okay, so as was brought up, there's a architectural tweak to tolerate or allow non-autonomous dynamical systems. We'll get there at the end. But for now, we're focusing on this system. Okay, so we're going to use an RNN. And just to briefly define what we mean, when I show this sort of top-down looking at the brain kind of graphic, what I really mean is a set of state space of G that is recurrently connected and iterating through time. And that these connections can be learned to generate the dynamics that we're interested in. And that after training, this dynamics is fixed. And it's implemented through the recurrent weights of the network. That's what we mean here. And so how do we get from a generator to our observed spiking activity, our bin.
David, are the weights from time step to time step are they yoked together, just like because it's unfolding? Yeah.
That's right. It's just an RNN
It's just an
that's
RNN unfolding.
with right. It's a tide weights deep network.
weights deep network.
That's right. Yeah. Okay. So well, we're going to have a little bit of stacking here to get from the generator RNN up to the observed spiking, and well, it's going to be the latent state, what I called the factors before, up to some inferred firing rates. So this is just a linear transformation, and then there's an exponential nonlinearity here to keep. So these are sort of like Poisson rates that then explain spike counts. So this is all, the sort of emissions model here is very standard setup for describing spike counts. Okay, and so we're just going to run the generator, and then there's this feed-forward set of layers to explain our spike counts, and so on and so forth. Okay, and that's all summarized here. We're going to fit our, I realize this may be belaboring it, but I just want to make sure everyone understands. We're going to fit our data that's high dimensional with multiple trials. We're going to get these abstract inferred instantaneous firing rates from a set of low dimensional factors. These factors are low dimensional because that's set as a parameter of the network. There's actually nothing about this technique that is dependent on having a low dimensional system. So that's also thinking towards the future. And that the generator now spits all of this out on a per time step basis, and the code is the initial condition. There's a whole other story of this is a variational autoencoder. It's actually a statistical model, but that's not central to the points that I want to make today, so I'm going to skip over it.
So are you saying the recurrent neural network weights are fixed? You're not like worried about figuring those weights out?
Oh no, so we will learn those weights. That's right, but then they are fixed. All I mean to say is there's this very standard train the system and then use the system for inference. That's all I meant to say.
So essentially, I mean, there was an older project I was involved in a long time ago. It tries to model a sequence like this with a hidden Markov mode, and which is similar in a sense, but there I assume some stochasticity in the dynamics, and essentially the states are discretized in some sense. So we're talking about discrete dynamics. But it's self-organized and learned, and of course there we don't really assume that it's autonomous, because there's always, you can couple the rest of the system with some sort of noise.
Right, so to
what's the difference?
To meet you halfway here, it's first difference is basically if the analogous version would be a Kalman filter, right? You have a continuous hidden state, and you have a Gaussian distribution over each iteration of that state, but that whole system is linear. In this case, it's nonlinear, and that's really the big win here.
HMM is nonlinear either. I mean, it can be nonlinear.
So fine. So, but this is with a smooth, high-dimensional dynamic. It's possible that maybe I'm reinventing something here.
It's not right. It's just a different limit of the same picture. But,
Okay. So the other thing is the noise model is a little different. I'm asserting a deterministic dynamics. Once you know the probabilistic code for the initial condition, that the feed-forward dynamics are deterministic. The recurrent dynamics are deterministic.
How do you choose the circuit?
It's learned. It'll be learned from the data.
No, I don't understand. Is it like essentially a complete a clique, basically, everything connected to everything else?
So yeah, so
some weights go to zero?
Yeah, so in this case, we're using what's called a gated recurrent unit. So it's a standard architecture in the deep learning community, and it basically is an all-to-all connected set. It's a complicated architecture. Oh yeah, it's all-to-all connected.
And some weights go to zero.
Well, who's to say? Maybe if you had.
It depends on what works.
Yeah, it could possibly be that way, but the idea is that you have this randomly initialized network to begin with, and then those random initializations are trained via optimization pressure to spit out the data that it's seen during training.
And how would you choose the initial states? What are they, the initial states?
Just random. This is randomized. The entire thing is randomly initialized.
All right, it's just to start the network, but
maybe
they are random at each time?
Oh, how do I? Oh, I see. Yes, now I understand. So you're talking about the initial state here.
Yeah.
So that is the encoder portion, and so it's just going to be the same machinery. So we have here. So this is now an autoencoder, right? Let me just pull that up here. So you put in your trials, your bin spikes. That goes through a different RNN, and at each time point, it's sort of sucking up the the bin spikes of that time point, creating now a code through time because an RNN has a latent state, and then that latent state is read out as an initial condition. Right? So this is really just a big autoencoder, and it's actually an enormous bottleneck here. Well, let me say it's a really tight bottleneck, right? We're trying to explain a sort of, if you will, n by t amount of data with a g-dimensional code, right? It's like a pinball or something, right?
You read out linearly the initial states from the
Yeah, that's right. We read out linearly, exactly. Okay, so this is the whole model. So it's deep learning methodologies. We have a negative log likelihood loss based on Poisson distribution of the output of the bin spikes. And so we could ask, well, what's interesting? Let's say we ran this, right? Let's say we trained this. Well, what's the interesting part of this? Well, the fire rates are interesting. That's sort of the first thing you would look at. It's basically saying, I'm going to deliver some abstract instantaneous rate that I believe on single trials is going to be extremely informative, or hopefully more informative than the bin spikes. Okay? You also have this set of low-dimensional factors, which is a low-dimensional representation of your data. And this is sort of an analogy to the rotatory dynamics that we showed in the motor cortical example. We have the trained dynamical system here, so if you had the wit to do it, and I have to admit I don't yet, you could study this system as a sort of encapsulation of a system that can generate the data that you are interested in studying. And then finally, the initial conditions, right? This is the code for your entire, you get one per trial. And so you could study those and make statements about those. All right.
I'm confused about the time, the factors. I mean, isn't this recurrent network have like the kind of history in it too, or like how do you? So
the factors will be temporal, but they're a linear readout. It's just the factors are a low-dimensional linear readout of the RNN state.
I mean, like the RNN has its own temporal dynamics too, right?
Mm-hmm. So it goes back to. Oh, there we go. Here we go. These are the factors. They're just a linear readout without any recurrent dynamic themselves of this generator state.
They don't have time? They're not in time, are they?
Their temporal dependence is only as a function of the temporal dependence of the recurrent neural network.
So they're like immediately reading out?
It's just immediately reading out the neural network.
But over time, the factors are a function of time.
Absolutely.
Yeah, you could draw a line across that way.
That's right. And we will. So we'll jump right to it. Okay. So the final statement here, if all of those details were a little too in the weeds, let me just say that what is LFADS? LFADS is a means of sort of denoising your data. But unlike, for example, just passing a Gaussian filter over a spike train, LFADS is different. It knows, like, in order to denoise, if you will, the spikes of a single trial, a single spike in a single dimension of a single trial, it's using all of the data of all of your trials to do that. Okay? And so that's embedded in the network through the weights, and that's what makes it smart and powerful. Okay? Because it speaks your data. So let's jump right to a synthetic example.
By the way, is there any guarantee this is going to work?
No, I don't see why not. I don't see why it would be a guarantee.
No, I mean, no, I guess I'm saying is how expressive is it?
So for the data we've studied, it's very powerful. I can imagine scenarios where it might break. So a scenario where it might break is if you tried to study V1 data, right? So V1 data, it's people have talked about and in the last few days all of these other dynamics going on in V1, but it's pretty well understood that V1 is driven strongly by a sensory system. And so there's no, in the setup, there is no input. So there's no way that this model is going to function well there. My thinking on the matter is that even though vision is like the paradigm of neuroscience, actually most of the brain is going to be a better fit here because the recurrency it's ubiquitous in everywhere, and you don't really have to motivate it too hard.
And do you need a trial structure in your?
No, you do not need a trial structure. That's right. It would definitely help to have a trial structure to understand what's going on, but you definitely don't need one. That's right. Okay, so let's just start with a very simple example, the Lorentz attractor. It's a three-dimensional system, and it's sort of visualized in its three-dimensional space there. So this is a ground truth example, and now you just see this system run its 3 dimensions through time. So we're going to make up some fake neurons, right? So this is like the factors, and we're going to make up this redundant representation by just taking random projections of these 3 dimensions. And now we can just artificially spikeify it using a Poisson process, okay? So when we do that, now we get a bunch of trials that are just spikes. Can we go back and learn this, right? Can we have learned the system, basically? And so let me just introduce very quickly a couple of other techniques. There is a recent technique, VLGP by Meming Park, GPFA by Byron Yu and John Cunningham and colleagues, and PFLDS, I believe this is also John Cunningham. These are recent, in the last year to 2 years. This is mid-2000s. And so anyways, when we train up all these models, and when we reconstruct, you can see just by eye that LFADS is doing a much better job.
Yeah, there's an assumption here that turning it into a Poisson process is not a very noisy channel in some sense. So there's some rate, the rate either change very slowly or some other regularization of the. So otherwise, it's just an HMM again. I mean, you have hidden dynamical systems, and you see a stochastic observation of it, and all you can do is reconstruct it within some precision.
So, so this may be an example of a well-known genre of tools, but I can tell you based on comparing it to other techniques that this one performs better, and that's sort of as much as I can tell you. And the reason why we're motivating these examples is because when we come to the neural data, you can lose your flooring very quickly, right? You don't know what you're looking at anymore.
In this particular case, the Lorentz attractor,
yes.
It's a 2 or 3 dimensional attractor at most.
Yes.
It's actually a low-dimensional attractor, and then you know, using, for example, Tucken's embedding theorem or something like this, that if you take enough points, you can actually reconstruct the dynamics. Mm-hmm. But these points is at least twice the dimensionality or something like this. Now, or D+1, I'm not really sure. D+1, yeah. And in this case, it's easy because we have a low-dimensional system. Once you have a much higher dimensional attractor, it becomes very messy.
Okay, so let's go to the next synthetic example. Now we have a 100-dimensional RNN that's randomly initialized to have chaotic behavior. This thing is a beast, okay? And so we're going to do the same story. We have here the underlying rates of the system. We normalize them and then spikeify it. And I challenge anyone to see this in that, right? So the variant is not relevant. It's a complicated system. And we just do the same thing. We train the system up on lots and lots of trials, and we compare it to other techniques. And again, you don't even need to quantitate it. You can just see by eye that LFADS is doing significantly better at this task.
The others seem to be low-pass filter, right?
Yeah, so in many, an assumption of the GPFA model is that there's a time constant that you end up smoothing things by, and so on and so forth.
And this is a one run, right?
This is one run, that's right.
So if you do multiple runs, maybe you get better, or...
So we trained on, I don't remember, call it 2,000 to 4,000 trials, right? We sort of picked the high end of what is done in experiments. And now you're just seeing the output of a single trial, and I would say that pretty much most of those trials are going to look like this one. Yep.
Sorry, so wait, by trials you just mean that an amount of time that's 2 to 3.
Just a random chunk. Yep, that's right.
Yeah, but you initialize a different initial.
That's right. You initialize randomly.
Yeah, so there's no sort of predefined.
No.
Right?
Yeah.
You're just running it.
That's right. Exactly.
And the training time you're saying is a few thousand times the length of this test.
That's, so yeah, it's like this times 4,000 is the data set.
And how long does that take to train?
Yeah.
So this a couple hours. So on real data sets, I'm not going to lie, this has all the benefits and all of the detriments of deep learning techniques. Hold on to your hats if you don't know how to tune hyperparameters. If you have a lot of data, you're going to have to chug through it. But, well, as you said earlier in the talk, deep learning is popular precisely because even if it's slow, at least it can handle these type of data. So it will live and die by the same criticisms.
So how large is the network?
In this case, I don't remember. I think it might have been order 50 or 100 units. Yeah.
So did you test as you make the network bigger and bigger? Do you get better results? Or?
Yeah, so it's an interesting question. I didn't do that particular study. I would like to think that it would improve. My guess is what's really going to improve it is more data.
But when you said how big it is that how many factors there were?
Yeah, so right, exactly. So I don't remember the number of factors. I would say these ballpark numbers are basically right. 100 dimension for the generator, 25 to 40 dimensions for the factors, and then the readouts are obviously dictated by the...
Is there a simple mapping at all of the RNN to actual connectivity?
Yeah. That's not the
very,
million
very
dollars.
degenerate
That's problem, very highly human.
So it's very likely highly degenerate and has
Yeah,
no
so
relation.
my thinking is that...
with the number of factors.
I don't know that you would actually, I used to think when I was putting these RNNs together that I was actually building brain circuits, and I don't believe that anymore. Yeah, I believe that what I'm doing is providing a rich dynamical framework for building dynamics, and there are yet to be...
I mean, I guess the general question is, it's a great predictor, but is it a testable theory about the brain?
I think there are plenty of things that will be generated as testable theories. But am I building, this actually is another question that came up in the review. How seriously should you take the connectome of this network? And I think the answer is, honestly, not at all, except for insights that you might derive from studying it, right? That is to say, if you know motor cortex generates a particular dynamics and you've made a system that can also generate that dynamics and you've done it robustly, then there should be something that's similar. But the nature of the connectivity could be completely different because the nature of the, if you will, the neural compute fabric is totally different.
Well, if you impose sparsity or something like this, you may find something like this.
So there's a lot of talk left, right? Let me just do this one.
Have you tried a non-gated RNN?
Non-gate, yeah. So you can do it. To me, it's a trainability issue, right? I've no doubt it would work just fine if you're willing to struggle through training it.
I don't see steam engine.
Yeah. No steam engines. Okay. So let's move on to some real data then. And this is data collected by Mark Churchland and Matt Kaufman. And again, I'm going to come back to this data set, the so-called the maze data set. For the purposes of basically the animal had to make reaches, right? And the purpose of the maze, they had their own agenda, but for the sake of this conversation, it's just a way to instruct a bunch of different types of reaches, okay? All right, so now we're going to apply LFADS to neural data, and so let's just take a peek at this. Is, for example, two arrays in motor cortex M1 and then premotor cortex. Both of these are associated in movement. So if you take a Gaussian filter at a single trial and just pass it over each channel, well, you have a parameter which is the width of that Gaussian filter. And so this is not LFADS. This is a Gaussian filter over each channel. And as you vary sort of the width, this is sort of the best you can do with that, right? So this is sort of what Gaussian filtering does to your data, okay? Now this is what LFADS thinks of your data. It was just totally nuts to me. Like when I first saw this, I was very surprised. Not the least of which is because oscillations pop out literally everywhere. Okay, so I went through some kind of like the stages of grief. You know, the first was like denial. Is that really there? And then I kind of ignored it for a while. And then I started testing it. Oh, it's really there. And I'm sort of at the acceptance phase right now.
What's the frequency? Is that meaningful, or is that like every time step?
Yeah, so this is, I think this is about a second of data. I'll get into all of that here. So here we have, I'm just going to show two more examples because it actually highlights. I've just used this red line to differentiate mode M1 from PMD. So here is the data, single trial, another single trial. These are the rates, the inferred rates, and these are the inferred factors. Right, so you can sort of see what this system thinks about that data. And you can see some slower type of dynamics there. This is, by the way, in a movement. So the reach is prepare the reach, it's instructed, go Q, go, right? So here is actually during the reach itself.
So what's the range of frequencies that we're looking at?
Yeah, so it's really stuff that's really fast to 15 to 40. right? And
that's beta and some gamma.
yeah, exactly. And so maybe we'll get to some analysis of that at the end. Okay, so I guess the point of this talk is a little bit of a show and tell to sort of verify or validate the methods. So the first thing I want to say is that everything I'm showing here is on cross-validated data. There's no training data on these pages. The second is that what validate means is sort of a strange thing when you don't have a ground truth, right? So the best I can do is show you examples and then correlate the denoised stuff to kinematic variables, right? To things that we've studied. So that's what I'm going to do, and I'm going to do a lot of it. But first we're going to take a look at some PSDHs, the bread and butter of electrophysiology. So each color is just a different condition. I've shown the smooth spikes. This is sort of what people do. But you can sort of average LFADS rates, and you can average GPFA rates as well. And so let's compare those. But on single trials, well, if you try to do a PSDH on single trial, it's almost nonsensical. It doesn't make any sense. Well, you can do that with GPFA, and you start to get some structure. And with LFADS, you see very tight structure. So, for example, this yellow condition has a lot of variation in the initial condition, if you will. I sort of revealed some bias there in the early part of the trial, and then it sort of tightens up and so on. And we can just look at a bunch of these, and you can see whoa, sorry. I made these a little too high resolution, so there's a lag that I'm not contending with. Okay, so you can just see that the structure on single trials here is sort of very stereotyped, and you could have really two experiences here. One is like, wow, that's real structure, and you've pulled that on single trial. The other experience is, wow, that is so stereotyped that maybe all you've really done is show the sort of the average, right? So what we would do then is show you that, let's regress to kinematics. So the animal had made these moves, and so we recorded the kinematics, and you can regress the firing rates to those kinematics. So if you do that with just smoothing, here I'm just showing a bunch of trials, right? For 1 condition, 2 condition, 3 condition. So you can see what it looks like with smooth spikes. And if you do GPFA, it's a little better, but you have to squint to see that it's a little better. And this is really the state-of-the-art single trial analysis beforehand. And now when you do on LFADS, you can see that whatever it is about those rates, it's really useful for decoding kinematics. And you can quantify this. Here, this is the R squared between the decoded kinematics and the actual kinematics. And the main point is that here in blue is LFADS. It's just much, much better. And critically, as a function of the number of neurons, we did a whole bunch of different LFADS runs where you change the amount of neurons used. You can just see that, for example, GPFA at 200 units is about as good as LFADS at 50 units or so.
What is GPFA?
GPFA is Gaussian process factor analysis. So it's basically you do factor analysis and then you have a Gaussian process model for smoothing.
Question. Are you regressing to the factors in LFADS or the inferred neural activity?
Yeah, it's a great question. And in different cases, I do different ones. In this case, it's regression of the factors. Thank you. From the regression of the factors to the kinematics. Okay, so we talked about that the code here is an initial condition, right, for each one of these trials. So here, I've shown you just a sample of reaches that the monkey made as a function of the reach angle, right? Just give you some sort of breakdown of that. And if we do a t-SNE visualization of the initial conditions, you can see that the t-SNE visualization is 2-dimensional. I guess it's 3-dimensional with a rotation in there. This t-SNE visualization recapitulates the structure of the reaches. So this says that the information about the reach is encoded in the initial condition. Okay. So now let's come back to the motivating challenge, which was, can we do one of these types of state space dynamic plots, except on single trials? And just to remind you, the single trial version was really super noisy. Well, when we do that now, instead of doing this analysis on smooth spikes, we do the JPCA. JPCA is the optimization to get the rotatory plane. We project our data into that, but now we use the inferred firing rates. What we see is actually extremely clean. And you can see that on single trials that these dynamics are showing up. And so everyone loves a movie. You can just take a quick look at that. So this is if you just do smooth spikes. There's some structure there. And this is if you do JPCA on the LFADS rates. And so there you go. Okay. So, yep.
In the single trial case, the initial states, the starting point for the motion is much more variable than it was in the average cases. So you were averaging the starting point as well as the entire dynamics?
Yeah, so the starting point is just the initial point of the, so everything gets averaged, including the starting point. Yeah, so then for the initial conditions, that code comes out of this encoder, right? That is the code of the whole system, and it's gonna have the variability it has. We assume that it got it right, or it got maybe too strong. We assume it encoded some structure into it.
Okay.
So another statement that you've learned the dynamics of a particular region in this particular, for this particular behavior of reaching is that you should be able to hold out a particular type of condition, and your system should generalize across that type of condition. So what we did here is we took all these reach conditions and we made, we just chopped it up into a bunch of bins. It resulted in 19 different sets of conditions. So what we trained is 19 different LFADS models with one condition, this reach angle, held out, left out, right? And then you say, well, if I really believe the dynamics, then I should be able to generalize to that reach angle even if I didn't train on it. And in fact, so that's just schematizing that. And in fact, when you run now JPCA on those models on the held out conditions, you see a very similar structure, and the initial conditions are nicely correlated, especially this first one, the held out versus when it was trained. Okay, so now we're going to transition to another LFADS architecture that we're going to address multi-session data, which has somehow taken on the term stitching together different data. Okay, so a practical reality of systems neuroscience is that there are limited measurements, and even as we move forward into this new era, people are still recording with single units, tetrodes, mu probes, v probes, etc. And wouldn't you like to increase the statistical power of your data sets in some intelligent way? Well, we're going to propose that LFADS can do this for you. Nevertheless, when a researcher goes in, an electrophysiologist goes in and studies area on day 1, day 2, day 4, day 50, they believe they're studying the same system. I'm assuming the animal has learned, right? So it's the same way here. You believe you're studying these same underlying dynamics, even if the day-to-day you take these neurons one day and this set of neurons the other day and so forth. So we're going to use LFADS to try to make sense of this. All right? So this is data from Eric Troutman and analysis from Dan O'Shea, who did a ton of work to get this right. So now we just have a center outreach task. Okay, it's still center outreach. It still reaches, but a simplified data set. And in particular, we're going to use I guess they're called mu probes or v probes. These are 24-channel linear probes that go in, and these two gentlemen recorded a lot, and so I exaggerate the example here just to show you. I mean, wouldn't it be nice to combine the 45 sessions that they did on different days, right? Okay, so in terms of, I don't want to belabor this, but in terms of the recording, they recorded over this, say, 6 millimeters in the precentral gyrus area. And so if we just look at some of the heterogeneity of the sessions, this is sort of target onset and reach, I guess. Oh, yeah, exactly, go cue and movement. You just see that there's variability, right? In the representation, you only have 24 units here.
So this is the same animal, and the probe was
That's right.
Inserted multiple times, or
That's right. Yes, exactly. Inserted. Sort of these experiments take a few hours, and you just 44 days. Yep. Okay, so here is just all 45. I guess it's 45. I don't remember 44, 45. For the upward reach, I just showed you single trials from all the different sessions, just to give you some of that variability. Okay. So now we have to augment this architecture. And the key is that we're going to have sort of readout matrices for each individual day. But the core architecture, the generative portion, the RNN, is going to be the same for all sessions. And the idea is that if there's any, for example, the units are different, so you need to have a little linear mapping from the generator to those different units on day by day. So the way we do that is we have, let's say we have two, this is 1 through S, but 1 and 2, right? So you're gonna have a different input matrix for that session that then goes through the common architecture of the encoder and the generator, and then back out to its individual matrix to get the random mapping. And for another session, it's the same story. Unique input matrix, common architecture, unique output matrix.
But when is the shared architecture learned? Is it learned with the same data, or? Yeah.
So so now when you batch up your data for a given batch, you have to pick a session, and then what you do is you say that you just set it up cleverly so that during the training, when you do backprop, you get gradients for everything, but you only apply the gradients for this session, say, to its readout matrix, its read-in matrix, and I should use this, and the model internals. And then for a different day, you exclude the read-in and readout matrices, right? So that's how you do it.
So what about what is this pre-session read-in? So that's a matrix that's also just plastic weights.
Yeah, so you have to be able to drive the system appropriately for this new data set. Right. So it's much more intuitive and easy to understand why you need a readout matrix. But there's a sort of, if you will, similar story on the input side.
The dynamics change significantly if you, for example, append session 1, session 2, and then use that as the training data?
That's an interesting thing to try. So it's a funny thing because that breaks sort of the single trialness of it, even though we're doing some hack with single trial here, because now you get single trials for session S, but you can read out single trials that didn't happen for the other sessions. But what you're suggesting is somehow even more radical, which is just tack sessions, single trials together.
It's like an ergodicity assumption.
Right, yeah, exactly. So we didn't do that.
So what important things are shared?
Almost everything. So the entire generative architecture is shared. What we built here is basically, yeah, exactly. So what we're saying is you're allowed, an individual day is allowed to drive the system differently and to read out, and to read out differently, but it still has to drive through the same dynamics. That's right. And so by virtue of the fact that we have different input maps here, you might think, well, geez, that's really dangerous, because you could really have S different dynamics. These generators are powerful. It could totally learn something nutty, right? And on each individual day. And so when I show you this plot, this is again just a low dimensional representation of these data. Now, instead of I've been showing you lines of single trials. Here I'm showing you each line is the condition average of a particular session for a given reach. So let's just focus on the orange there. Let's just call that the upwards reach, right? That's the conditioned average dynamics for session 1, session 2, session 3. So you can see that the system found a common underlying dynamics that explained all of those different sessions of data, despite the heterogeneity that was shown for that upward reach in the early setup.
So I want to comment, this is highly discretized. Right? You have
Excuse me?
essentially 5 different states here or something, no?
Highly discretized. I'm not.
In the number of colors, at least.
Yeah, that's the different reaches. That's the reach conditions. Okay, so these are just. I just showed you. This is just a 3-dimensional view, and then I put it forward here. So.
What's the CIS coordinate?
Yeah, that's a condition independence. So you just sort of average over everything, and that tends to be orthogonal to the JPCA rotation. Okay, so now we can look at this at single trials. So here's the hand reaches to begin with. It turns out that orange was down and to your right. But if we can do this is going to be kinematic decodes again, right? And the idea is to demonstrate the additional power that stitching together these data sets provided. So smooth, GPFA, is a pattern here.
And that's smoothed over all trials, right?
Oh, no, I'm sorry. This is
single trial.
Single trial. This is you Gaussian smooth the spikes, yep. So now with single session LFADS, it's better, but you only had 24 channels here, so this is still a little wonky, right, on the individual trial. If you go, that is to say, you train LFADS on one session only, right? And now, when you do multi-session, it really cleans up the factors here, right? It really cleans up it. And I just want to highlight this difference is large, and this difference, this improvement is also large. And you can quantify that. And you can say that the R squared, again, R squared between the inferred kinematics and the actual kinematics, you can see that for every single data set, there was an improvement by combining the data sets in this way, right?
I hadn't noticed.
Yeah, sure.
So this is monoclinic, so you have sequences of some of the cells responding to muscle activations, right? So you can have sort of pretty unique combinations for different trajectories. There's no symmetry there. Maybe that's how this is working. If you were to take a system like the head direction system, presumably you wouldn't recover the same head directions in each in separate sessions. You might have to rotate it to get it to match.
So the way that I interpret it, I'm not entirely sure what you were saying, but to the degree I understood it, to the degree I understood it, what's going on here is that, if you will, the wiggles have correlations across sessions, right? And so if you have any sort of optimization on simplicity, which we do, there's regularizations in there to keep the system as simple as possible, then yeah, the system should try to make us, the optimization should try to make a simpler generator that takes advantage of the correlations and the wiggles across sessions. Yeah.
As a biologist, I want to ask, would it work between animals? Does each animal have its own internal dynamical system?
I would, right.
I would guess that it's the same between animals.
This is at the very, very top of the list of things that I want to do next. Because there's a few technical problems. I'm not sure, for example, a linear readout is going to get it done between animals. But one would imagine that some kind of topological preserving, but warping and shifting and things like that, you might still have the same dynamics aside from that type of.
But one is low dimensional, one is very high dimensional.
Which one?
Never mind.
But again, you're going to have different cells, of course, in different animals, and what's going to relate the dynamics?
Well, the belief is that if there's a common dynamic and all that's being done is, say, just let's take the easy case of just rotations and shears and things, then you
should look some invariants, which should depend on the way you.
So it's an open question, and if I was going to put money on it, I'd say this is going to work, honestly. Yeah.
So, by the way, this is all M1?
This is all these. I think so. Yeah. Yes.
And have you tried premotor, and have you seen any differences?
Yeah. So for these V probes, no. What we've done premotor work is with these Utah arrays, where we always have premotor and M1. Yeah.
So what is this hand? What is hand?
Oh, hand is the actual kinematic reaches that the animal made. So yeah, we're taking the neural data and decoding. Just because the animal has variations. Yeah.
How many trials in a single session? Because before you said you trained it with like 4,000.
Yeah, so I want to get back to you on this, but I'm pretty sure the answer is order 200 to 300 on a single session.
So here you've got fewer trials, and then you're increasing the number of trials, but you're also shifting the input and output.
Yeah, so you're combining you're combining your data, but you're not entirely certain it's going to work out. Okay, so. Peter.
So this is beautiful analysis. All segments like that are going to have a flat in it.
So what do you think is potential? I really talk about this at the end for giving us deep insight into how the brain works.
The potential. So the first answer is going to be, if you did analysis X before, do LFADS first and then do analysis X. Right? That's sort of it's not a very satisfying answer to a theoretician, but I think to people who are in the data all day long, if you can get like dramatically better decodes to kinematic variables or to any variables that you study, the chances of you making meaningful scientific statements is going to be, I think, dramatically improved.
So how much computation can it be done in real time for BCI?
That's another thing that is second on the list, actually maybe third, but yeah. So there's a version of this. So this is like a Kalman smoother. I didn't go into the details, but you get to see, the method gets to see the entire trial, and you have to disable that, and it will be the power will be decreased because of it. But real time is nice, right? Okay, so I'm going to push on. I don't really know how much time we have, but I'm getting towards the end. Okay, great.
You have lots of time.
Okay, great. So we have here, I'm going to show you some movies, and now what I realized last night in preparing this, in the final preparation, that I was missing a slide here. So I showed you these really nice representations where all the different days' data looked really similar to each other. And that's in the stitched model. What I don't have the slide for, unfortunately, is to show you that the individual LFADS models, one model per session, it does better than GPFA, but it's not overwhelmingly good. So there was really an improvement there. So in this movie, I want to try to whoa, that's definitely not where I wanted to go. Sorry. Okay, so we go forward. Does anyone know how to pause on Keynote? That's what I'm asking. Spacebar? No.
You have to take it out of display mode.
Yeah. Okay. Let me do that. It's worth a hot sec here. No, geez. Okay. You know, I'm just going to play it, and we'll see what happens. All right. So the first thing I'm going to show you is GPFA, another technique. A single session. And
what is a single dot? Just tell us ahead of time.
Yeah, exactly. A single dot is a single trial. The color is the reach direction. So what I want you to see here is that these low dimensional projections are not particularly inspiring. That's GPFA. Now for a single session in LFADS, same thing, single trial, and you start to see more structure. But again, it didn't have that nice rosette structure that's a hallmark of JPCA. Now we do a single trial, a single session of LFADS, and you can see you've got the nice rotation here, because why not?
You got the Lawrence target.
So by combining the data, I have to get through this movie, otherwise I can't stop it. So by combining the data, you see in the movie, you got a really cleaned up representation on the single trials. So now the next step is here, I'm going to show every single session, 42 of them just to fit them. So this is GPFA, again, not much structure. So this is what I really wanted to highlight. LFADS on different models, yeah, it's got structure, but it didn't find that commonality by combining and stitching. So yeah, okay, there. And now finally, the stitched model for all the different sessions, and you can just see whoa, there's lots of structure there. And again, I want to come back and just highlight, because the stereotyped nature of this is so strong that you would think that you've lost information. But I just want to highlight that these kinematic readouts are dramatically improved by doing this.
Sorry. So it seems that smooth GPFA, etc., they don't really assume a dynamics or try to extract the dynamics. So I was wondering why you're using a pretty powerful and complex network, whereas you could try first linear dynamics or other methods.
Yeah, it's taste, right? I mean,
taste. Yeah. I mean, the simpler models are easier to interpret, right? I mean, it's not only test.
So first off, there are people. Okay, so maybe it's uninterpretable. That's a reasonable criticism, right? My take is that we know the brain is nonlinear. We know that the brain has contextually different dynamics. So just embrace it.
But it's data analysis, right? So for data analysis, the goal is interpretability.
Yes.
So you may need a nonlinear model, but it may not have. I mean, looking at this trajectory, it doesn't look like you need a 100-dimensional recurrent network to reproduce them. That just
I would come back to I'm building for the future here, right? I mean, I guess I'm just not sympathetic to the complaint. I mean, sure, go ahead. You could swap in a linear model into the generator. You could do it, but we already know what's going to happen. It's going to do reasonable job on linear things.
Piecewise linear.
So you could do piecewise linear. That's right. And Scott Linderman has been pushing this forward in the last year or two. In that case, there are more difficult optimization aspects to it, though, because there's a discretization in the latent state.
The future is not necessarily getting more complicated. The future is getting more simple. That's how you progress in science. Sorry, but that's my philosophy.
So look, this is a point in time, and we're evolving. And it may well be that there will be decoding techniques to look at your complex chaotic network and be able to read out things from it. That'll give us understanding. So I don't think we should use that as a criterion right now.
So I guess what I would say is, concretely, what does LFADS give you that right on the table is useful? The inferred rates, the factors, the initial conditions, and what I'll show you in the last segment of the talk is these inferred inputs in the non-autonomous case.
A fair comparison for performance would really be a model that assumes a dynamic. So this model do not assume a dynamic.
No, so I totally disagree with that. That is a fair comparison, but another fair comparison is just take whichever model you think is the best one and give it the best crack that you can to do its job. And that's what I've done here. We spent time, we talked to all the authors to make those models work as best as possible. So it's just that's another fair thing to do.
I think it's a great idea to do this generative model. And my criticism is about the type of network that we use.
Sure. We can swap. The code is available. If you want to swap in a linear system, feel free. It's there.
Well, the question is how seriously you can take your dynamics. And I believe that there's not enough. I mean, anything, a linear predictor which is changing in time, autoregressive HMM as we call it.
So I've shown you that we can decode ground truth of a chaotic RNN in simulated case. I've shown you that we can do better decodes. I don't know what else I can do, right? What else do you want?
Counting parameters.
This is a great conversation. I'm going to move on so we can finish up.
Right, good.
Okay. Okay, so behaviorally relevant inputs from single trials. The last part of this talk is somehow the most speculative, but I also think very interesting. So the motivation is we all record from areas, we meaning my experimental collaborators, where you know the system is getting inputs from all over the place, right? So this assumption that we're modeling autonomous dynamical systems is obviously incorrect. And what we've shown is that for places like the motor cortex, in certain behavioral paradigms, it is not grossly wrong. But there's caveats. For example, if I build a system and I allow the variability of the trials to be stuffed into an initial condition or into an input, now all of a sudden there's a identifiability problem, right? That's an issue. Another issue is if there's noise and I let, like in a Kalman filter innovation, I just let that noise come in to fit the data better, then I don't know can I interpret that? I have to be very careful, right? So, but we're in a world where we know these inputs are coming into the systems we study. Let's try it and see what happens. Okay, so what that means practically is changing this model to include an input U. And so now fully specifying this system requires learning the dynamics, an initial condition, and U. And I just want to point out explicitly that if the complexity of U is not limited in some way, either low dimensional or the complexity of the wiggles, this is no constraint on your system. I get that, I acknowledge it. And so we'll show you what I hope are systems where we've constrained it enough. Okay. So now the LFADS architecture has to be updated. I'm going to be very brief here. But basically, the generator now needs a controller. It needs another system to tell it when an input comes in, right? And so that controller is going to spit out a u of t at every time step, right? And it gets to see the input data through some encoders, and it also gets to see what the generator outputs, right? So if you're familiar with Kalman filter innovation, this is very similar, except that it's pushed into the generator. So the inferred input literally is input to the generator. And without really taking some real time to explain this, that's just going to have to suffice. Okay, so
what mu and sigma are two different types of control.
Oh, so mu is so it's another statistical code. So there's a mean and standard distribution on noise, yeah, in sort of keeping with the variational autoencoder architecture. Okay. So now instead of thinking, I guess another way to say it is, before we were saying we have one vector that explains a time series of data. Now there's this sort of, if you will, allow me, a strange outer product. You have this initial condition that's for one time point, but a high dimensional vector, and you have this 1 by t vector, or maybe low dimensional by t vector, and it's the combination of these two things that define the dynamics of a single trial. Okay, so let's again fall back to synthetic example. Here we have, excuse me, I jumped way too far. Okay, we're just going to build an integrator. Take an RNN, use backprop, build an integrator. It takes white noise, and it outputs a bounded integration to +1 or -1. In green is the target, in red is the output of this system after it's trained. So now, of course, we want to say, can we identify the input, right? So after training, this is what the spikes look like. After training, I've just shown you 5 of the instantaneous rates. You can see it's doing a pretty good job. And here, for sort of an average example, in black is the actual input to this system. And the inferred input by LFADS is in cyan. And I would argue if this was the performance that was given for biological data, people could actually use this. This would be a meaningful signal to study. Okay, and so this is a 0.3 R² example that I showed you over here. Okay, so now let's move to some data. And here, Sergei Savitsky actually created a task that is perfectly set up for this. So animal is going, it's a BMI task. Animal is going to, it's too strong. Let me just explain it. There's a cursor, and then the animal has to reach towards a target that pops on the screen. And so for many reaches, it's just unperturbed. But on occasion, the screen updates the location randomly of the target. So there's jump right and jump left. So we have 3 conditions here, recording from M1 and PMD again. And now there's ups and downs. And so, there's up reaches and downwards reaches and the associated perturbations. Okay. That gives us 6 conditions, 4 of which are perturbed in ways that are unpredictable to the animal. All right, so here is an unperturbed M1 PMD, M1 PMD, unperturbed, perturbed left, perturbed right. I could make up stories, but I can't see anything in that personally on single trials. This is the hash. Okay, so when we run LFADS, I'm just showing you some more data. These are what 8 trials. This is what this looks like, sort of. Okay. And now we examine the inputs. We used 4 inferred inputs here. 4 can be motivated because there's a sort of the reach direction, and then there's the trial type. There's the trial type, and there's the perturbations, and there's also some LFP in there is getting sucked up. Okay, so if we focus here, the inferred input, now I've color-coded this. Perturbed left is blue, perturbed right is red. This 4th dimension of the input on the trial average definitely encodes which way the trial is going to go. Right? And so I'm sorry I didn't tell you something basic here. This is the target onset, and then later on in time, this triangle is the perturbation onset. And so later on in time, we see at the triangle, there's clear separation in this first input dimension that dictates what the perturbation should be. And it comes in at the right time and it exists first off, and it comes in at the right time, and it sort of separates by condition. So when you look at the single trials, the inputs are messy, but they separate. And on single trials, if you do a t-SNE plot of that t-SNE plot of the little time segment around the perturbation, you can see the perturbed left and right and the unperturbed trials here. Okay. So there's one tiny more segment, one little more segment, and I'm done.
Before you go to the oscillations, I think that's what Les said.
Yep, that's right, yep.
So here's a question. I mean, we saw how manifold embeddings, our real neural data really seem to be great. You know, this is ELO. Could you use that same technique on your recurrent networks, generators, to see if there's a hidden a manifold there?
Sure, absolutely. So right now what we've been doing is using techniques that were sort of already derived, used sort of practice in the literature, such as JPCA or whatnot. But yeah, in any technique that's going to help identify meaningful manifolds could be used for the generator. And on top of that, we do have the factors, which is some version of that. Okay, so coming back to the acceptance phase of seeing oscillations in the data, here we have a reach, and you see this sort of beta, like a 15 hertz or so, in the preparatory period of the movement. And this is actually really well known. It was new to me, but we all learn things. And so the spikes are synchronized to the tops of the LFP oscillation. This is true in monkeys and in humans. So we wanted to study this, but even you might think that you don't need single trials to do this, actually, but you do, because if there's any, first off, oscillation is a very loose term, right? I mean, these things are wiggling all over the place in terms of their frequency. But on top of that, even if they were perfect oscillations, if you don't get the phase right, you're just going to average everything out, right? So that's what we mean here. And so let's just show the data. Here's a single trial. Again, M1 and PMD. This is much stronger in PMD. And here's the local field potential visualized. So what we're doing here is we're showing you, this is on the channels, right? So this is a Utah array with the 2x2 channels, and we're just aligning them.
So one question. You see these very narrow vertical lines in your rates, right? Do these correspond to something that Sonja Grün long time ago called unitary events? So are these really kind of population spikes in sets of neurons that go on almost together?
So what I'm going to show you is that they correlate tightly with LFP. And beyond that, I don't.
But no, I think the way to think of, I mean, the inferred firing rates are the instantaneous rates. Right. But with the Poisson generator, you're not getting these spikes out. So you're only
seeing. If you have a firing rate that goes up like crazy in such on this vertical line, right? It seems almost. I mean, what worries me if I see this is that compared to the firing rates, these rate changes are very fast and very steep.
Well, they're definitely. So first off, they're definitely there. It's not on the screen, but we did control it.
So my question is, how is this reflected in the data? So does this really mean that there are subsets of cells that go almost?
Yes, that's what it means. That if you were to sum over this vertical dimension, it would start to look a lot more like what you're seeing there. It's exactly what it's saying.
The histogram.
Yeah, exactly. So it's really there, and we just don't really focus on it. Okay, so what we did here is in black we correlated the spikes with the LFP, right? And that gives you here those wiggles. And then with LFADS, we took the inferred rates from LFADS and we correlated them. And you can see these are just for different channels, channel 13, channel 19, there's a very, very tight match, which means that when you see a wiggle here, it's actually, because I should have said that the multi-unit array actually records the LFP as well, right? So you can compare those things. And now when you shuffle trials, everything gets washed out. Okay. So I just want to point out, this is a sort of interesting caveat here, which is we couldn't do this without the inferred inputs. So that is to say, the inferred inputs are providing information to these trials through time that was necessary to make this correspondence. Of course, with an RNN, you can fit anything. But on the held out trials, do you have any data there, anything to say? And so what I take this as is that even though you think of an LFP as this really sort of simple signal, because it's low dimensional, it's actually really complicated in time. And so so what this argues is that, well, what sort of the lore that's said is that these LFPs are somehow involved or a byproduct of communication between different areas. And so you can't actually model it in the sense of modeling it dynamically.
And so this is 5 to 10 volts.
This is closer to 15 to 20.
Yeah, that's beta.
Yeah. Okay, so I'm done. So where are we going? It seems pretty clear-cut you'd want to have a calcium imaging model on this, so that would change the distribution. Instead of having Poisson counts, you would have Gaussian or something. Applications to brain-machine interface seem pretty clear-cut. I'd love to just like see what happens if you take a bunch of neural pixels probes and you try to do this stitching. I mean, that would be really super interesting. And then finally, reconstructing dynamics across animals, right? I think that's also very interesting. And so thank you for your time. That's all I got.
So my question is I think that this is very interesting, and you could say that the model is too complicated, but I mean the stitching I think is very, very interesting. But the question is a little bit, how do you interpret this as a computation in this brain area? And it almost feels to me the behavior that you're studying is too simple, because you want really to show that all this learning that you're doing in your artificial network, that you really learn a repertoire of interesting behaviors that are, I mean, my question is, can you somehow tie this to information processing? Can one somehow say what it does in terms?
Right. So what I would say there is, first let me start with the caveat I made before. I think we're building abstract models of real dynamics. And so I don't hold much hope out for connectomics-like project.
This is not my question.
I know, yeah. So just stating that.
Yeah.
Having said that, if you had enough trials and enough varied conditions and so forth, then this is going to learn that. It's going to learn that system. So it will be a very meaningful, though abstract, model of the dynamics that are ongoing in a particular region. So the question then becomes, can a very accurate abstract model of dynamics be a useful scientific tool? And I think it can be. I think you can ask questions about, for example, perturbations, right? You have these dynamics, you think, well, I believe if I perturb in this dimension, then this type of dynamic should happen. If that holds up to be true, then yeah.
So there's a precedent here in dynamical systems theory. So you saw the Lorentz attractor. So what they have proven, that with a simple embedding, in other words, you just take one variable and embed it into a value at t versus value at t minus tau, that from that trajectory you can infer the attractor. You can infer some fundamental dynamical property of a very complex underlying system. And so this is potentially what you want to bring, what you want to extract from the brain.
That's the Hawkins'
What was that?
Hawkins' theorem.
Yeah, Hawkins embedding theorem.
He's been going. One sec.
Your loss function depends on the input spike train and output spike train.
Yes.
So, what is the law? How do you define it in a way that's so it's differential to weights and everything?
Yeah. So this is the variational autoencoder. So it's a negative log likelihood loss with a Poisson output distribution, and then there is a stochastic hidden layer, right? That's the codes and also the inferred inputs near the end of the talk, and that those use to back propagate through those stochastic layers. You use the reparameterization trick, whereby the stochasticity is the difficulty of that is ameliorated.
Yeah, so I want to return to my question of what is the minimal dimension which actually makes sense. And one way of testing it is to keep out some of the neurons outside of your model as some sort of test set, and actually try to infer their behavior from the other neurons.
So we did that.
And at some point this should collapse. I mean, and this will give you some sense of the minimal dimensionality of the problem.
So you're saying that as I decrease the number of dimensions in the generator, the fit to the held out units should deteriorate. And that tells me...
Well, so there should be some sort of a knee in this fit, which should tell you something about the truth. Whether I can actually trust anything you say.
I see.
And again, it's very closely related to the work we did in 95, 96 with Abeles and some of my students with HMMs. And we actually had, very similar areas in monkeys. We did it in the no-go phase, in the delay between the go and the no-go phase of a delayed response experiment. And we actually found wonderful state transitions. And the way I trusted it then only when we actually could cross-validate it on an held out cells.
So
actually, we even did it across animals. But
so I take your point about studying the dimensionality with respect to held out neurons. But we've done a lot of controls here, and I don't know if I've convinced you, but at the very least I've convinced myself.
It's actually very beautiful now. I mean, the whole thing is nice.
Okay, so we just to enumerate it, obviously cross-validation for training, so everything is held out. All the data shown is held out data. We did held out conditions, right? And we also did, I didn't show it, but we did the held out, the so-called co-smoothing. The problem is I didn't show that result because it's really hard to know what one bit of log likelihood, I don't even know, I have no idea what that means, right? But anyways, we did that too.
So here's the question about generalization. So you have data from the monkey doing, 8 inside out. If you had additional data from the monkey just moving his hand around, right? I mean, not necessarily in and out, but around. Do you think that your model trained up on the in-out could do the kinematics for the more general movement?
It's a great question. So I can't answer your specific question, but I can paint a sort of one parameter family of answers, if you will. If you have two different data sets that are totally different, and there's not much data associated with either of them, then the chance that an LFADS model trained on one of those data sets has anything to say about the other is essentially zero. Now you move that forward to a scenario where you have basically really rigorously tested the behavior of, let's say, the arm, since that's what we're talking about. You have weights and it goes up, it goes down, it goes left and right, then yes, you will. That model should be taken very seriously. And I think this comes to, at the high level, that these deep networks are very good at interpolation and not so hot at extrapolation, right? And that's what it boils down to.
I'm curious about how the condition labels work. Like if you gave it the center it reaches but didn't say which reaches or which target, how does that change things?
Oh yeah, I'm sorry, I should have said at the beginning. There were no condition labels here. Not any example at all. This is just pure auto-encoding of data.
So the autoencoder model is interesting because you assume that there's some mechanism that generates this dynamic, and then you perturb, right, and you get this U. But you had a very different data set there where the animal was reaching. So, it could be that if you analyze the perturbed data with just the autoencoder, right, you'll also get something out, right, that says left and right. That's not necessarily
we
from
tried it.
you, right? Not
Right.
from the input. So, I'm just wondering how do you?
This comes down to it's an ill-defined thing to try, right? So how do you reason about it in a scenario where that particular data set was almost perfectly constructed to make the point?
Right.
And if you're outside of that setting, then how do you actually trust something?
So your U could turn into just another dynamic in the sort of center out reaching.
That's right. So in general, the issue is, I think, unsolved. I don't have an answer for you. In practice, there are things you can do to feel more confident about it. Number one is only have one or two inferred inputs, and then you know it can't describe everything. There is also regularization pressure on that inferred input. I didn't go into it, but there's a regularization pressure to deliver no information. It's a KL penalty to an uninformative Gaussian. So that's all on there as well. But I take your broader point.
Well, it's just how you formulate the question that you're asking.
Can you comment on the application to BMI?
Yeah, sure. So the idea is that there's I guess a more recent line of thinking that if you had a model of the dynamics of your system, then you would be able to use that it's like a prior on, I mean, you're being very informal here, but it's like a prior on what happens when you actually go into a BMI mode. So if you could actually model that, so right now, this may be more detailed, right now the standard model is a Kalman filter that assumes that the kinematics drives the spiking observations, which is, it's flipped, it's wrong, right? I believe the right way to do it is that the spikes drive the kinematics. And so now you have to develop a model for those spikes, and that, just from taking a few days of data, may not be very rich. But if you had a model like this, we have to get the data for this somewhere. But if you had a richer model of the of the neural dynamics, that would definitely inform the state model in this basically nonlinear Kalman filter.
Yep. So you have your data from premotor and motor cortex. How are you thinking about this control input? So I mean, if you, for example, would model the dynamics in the premotor area, and you try to infer your control input in the motor area, would you think there would be a relationship? Or
yeah, that's a good question. Could you use those two? Basically, if you have an input and you have an output, we haven't done it. We should try something like that and see what we can get. Yeah.
So have you tried, instead of using spikes, using the LFP, since you have that, and just trying to do the whole, generator based on...
We haven't tried it. It's an interesting thing to try.
It has been shown that BCI can actually be, there's a lot of information there, as you've already shown, that is in a sense, might be telling you something a little bit different from what others have inferred from the spikes, right?
Sure.
And by the way, if I stop there, try the EEG.
Now you're getting radical.
And here's why. I mean, I saw Fritz's. I mean, this is talk about radical. Okay, here's somebody who has gone against the received wisdom of a 50-year history in the hippocampus, saying that there is no spatial organization, that it's all basically the salt and pepper, right? But he's showing that nonetheless you can extract information from this very crude oscillations, just in terms of the phase shifts and phase differences, that allow you to do the decoding for position. So there's actually a lot more information there than we thought, right? And
sure, absolutely.
And so, it's if it works, it would be unbelievably important. And the reason is that, putting electrodes in is very invasive.
Exactly.
Right?
Yeah, that's
so
right.
If you can actually extract more information from the EEG than other people have, then that has a huge implication.
TBD, that's hard work.
So for the session stitching part,
Yep, sure.
You're extracting like the common component across all the different sessions. And so there's obviously a residual component. And I guess how do you think of that in terms of like the variability that's going on? And if you could sort of subtract that away or somehow differentiate it away from the common part, can you somehow relate that to other behavioral variables that are changing from day to day? Or I mean, how are you thinking of that?
Right, so I think there were two ideas sort of conjoined there, right? So the variability of the trial is going to be stuffed into these two matrices, and really the powerful variability is that the input matrix can determine how the system gets driven, right? And that can be arbitrarily powerful. I mean, these systems can learn, if you have S sessions, they just learn S different dynamics, right? So that could be pathologically powerful. So that's where, it's the initial condition that would be expressed as the code that is differentiating between session. And if you wanted, now I guess the other piece of this is that if you wanted to try to study somehow non-stationarity, for example, learning or drift in representations.
I guess I thought it was just like the readout part that you're changing from session to session. Like,
yeah, and an input part.
You're saying there's I see it, and the input. Okay.
That's right, yeah.
But then what is that? I mean, what's changing from day to day?
Well, so the way I see it is the readout is what you've measured is changing, right? So you believe that there's some underlying system that is low dimensional, otherwise it's hopeless anyways. And one day you're getting this particular random matrix readout of it, and another day, to be imprecise.
Why? Why isn't it the same from day to day? Recording from different cells.
You're recording from different cells.
Oh, I see. I'm sorry. I didn't
yeah,
get that case.
yeah.
The array is kind of
it's a different electrode.
That's right. So in this case, these are V probes where you actually take them out and put them back in. Yeah. But you could still do the same thing for Utah arrays, where it's chronic, and then you hope that there is shift. I mean, in BMI, it's an enormous headache, right?
So if you didn't believe in photon spiking, you could have a mixture of faster processes, couldn't you? And that would be your underlying model.
Yeah. So I guess I sort of agree in half. So the issue is you could forget the Poisson output. You could have some biophysically deep.
Because now you have to sort of pick a window that seems to work on the data.
That's right. So there's a bin size issue, right? So that.
But it could be that it's just a mixture of a small handful of processes would be ultimately a better model.
So I guess there's a philosophical point, though, which is I believe I can identify a low-dimensional sort of smooth dynamics. I have in fact, I would bet against it with a lot of money that I could actually identify a dynamics whereby I'm spitting out spikes at each point. Right?
How much money?
Well, I mean, you could always fit it, but will it generalize, right? I mean, the actual spike generation process would require understanding an enormous amount of detail.
You are seeing gamma frequencies.
But it would require understanding an enormous amount of detail about the biological circuit that is essentially prevented from us, right?
You vary the rate of the Poisson processes continuously.
That's right, yeah.
But within some limits, I guess. So it's a band-limited signal.
Yeah, so there are no, so with regards to time constraints, this is even though we think of these dynamics as continuous time, this is a discrete time system. So whatever the Nyquist rate of your sampling is.
If it's arbitrarily varied, then it's anything. It's not a Poisson process.
Right. So so in practice, that won't be what's going on, right?
So if you take your d-dimensional factors, each of which is a function of time, and so you could think then of the system evolving in those coordinates at a uniform velocity, right? So in some sense, the factors are a dynamical system, and what you've done is factorize the complicated dynamical system into this part that you're pulling out that you're saying is session dependent or something like that, and this other part that you're saying is universal. Have you thought of that decomposition of the two dynamical systems? Because you're kind of calling the factors these matrices that you stick in the front that go before the dynamical system. But really, if I'm understanding what you're saying, really the factors are just another dynamical system that you're wandering through at a uniform velocity. And so you've factored that out of the other dynamical system.
So the way that the stitching works is that the factors, which are a linear readout of the generator, so the generator could be a million dimensional and you're getting 10 factors out of it, right? So those factors are common across sessions. That's the common part. And then what is no longer common is then from those factors you have to convert to the different actual instantaneous rates that explain the spikes on that.
Oh, so it's after you do the factoring.
That's right, yeah.
I see.
That's right, yeah.
So Andy Schwartz did an experiment which I think is very provocative. You know, he's been doing this decoding for a long time, and he's one has this movie you see all the time of a woman picking up the coffee cup for the first time, right? Robot arm. And so he had a graduate student who's, every day they have to calibrate it, by the way, because it drifts, even from the same array. And one day, one of the students said, well, look, what if we just randomized the tuning curves? Or reverse them, let's try this out. And of course, Andy said this is crazy because, you're throwing away all the hard-fought detailed calibration that we have here. And sure enough, when the monkey first started out, it wanted to have its arm go this way and it went that way, right? But within the same session, it recalibrated itself. So the monkey has the brain in the motor cortex has some adaptability so that it can reinterpret, the pattern it needs in order to get its hand to the right endpoint. which means that you have potentially the ability here to look at learning. But it's really and it's within one session, so you don't have to train for 3 months.
Yeah, so that'll show up in the.
I mean, and that might you said that learning is impossible to study in monkeys because it takes so long. But if this is a form of adaptability in the motor cortex that you can study in one session.
No, I didn't say. I said. Yeah.
But in any case, that's another dimension to study. Is how
But in that
mechanistically.
Can I just make a suggestion? I can see the room that you guys can't see, and I can tell people are hungry. So why don't we have another question and then close it out?
I was just commenting on this scenario where probably the factors would be the same, and it would be the output mapping would change, right? So,
So that type of variability would show up in the codes, right? It would show up in the initial conditions, and you could study that as a function of day or whatever, right? And then it would be some summary of the drift, even if it's not mechanistic, right?
Okay. So one sad announcement actually. Flip Service just mailed us, and he fell ill and he cannot make it. So this means there's no more talks anymore. So this means we could now wind down the workshop. We had a very good discussion yesterday. Who believes it's raining in the early? Who doesn't believe? Who believes it's not raining in the early afternoon today?
the sun is out.
Exactly. So whoever believes that and wants to take a walk in the afternoon, so I would be up for it. Let's have a good lunch now, but maybe at 2:30 or so we could meet and go up the fire trail here and take a little walk and continue discussion.
2:30.
But unfortunately, this winds down sooner than we expected. I mean, I think we enjoyed it tremendously. I hope you as well. Thank you for all the participation and all the talks and the great atmosphere.
And thank you.